This video is on photosynthesis in plants. It's a detailed breakdown of the process of photosynthesis, including all of its metabolic details and some information about the anatomy of plants too. In this video, I'm going to deal with plants in the sun. I'm going to summarize the process of photosynthesis. I'm going to discuss organs involved in photosynthesis. I'm going to discuss the light reaction, the Calvin cycle or dark reaction, C4 plants, CAM plants, and then I'm going to make a comparison of C3, C4 and CAM plants as they relate to photosynthesis. What is the currency of life? All interactions between living organisms and their environment involve the exchange of energy. Energy is the single most important currency of life. No living organism can function without the use of energy to drive its metabolic functions and processes. One could argue that life also uses information as a currency to exchange for the purposes of replication, reproduction, and communication with other organisms. However, without energy, information cannot be exchanged between organisms. What are the two kinds of living organisms with respect to their energy intake? All organisms gain or get their energy from the sun, with the exception of certain bacterial species and their associated symbionts that may derive their energy supply from deep water thermal springs and chemical processes deep underground. There are two kinds of organisms based on how they obtain their energy supply. Autotrophs, which produce their own food using energy derived directly from the sun, for example all plants, algae and cyanobacteria, or heterotrophs that derive their energy by consuming other organisms. Heterotrophs might include herbivores which feed on plants, carnivores which feed on herbivores that consume the plants, omnivores which might feed on plants or animal matter. These consumers might be referred to as primary and secondary and even tertiary consumers depending on what level they feed on the energy table. So what is photosynthesis? Our focus is on plants that derive their energy directly from the sun and fix it in molecules such as carbohydrates in a photochemical process called photosynthesis, which literally means synthesis by light. The chemical process in its simplest form is as follows. Carbon dioxide plus water plus energy from the sun in the presence of chlorophyll is used to restore energy in the bonds of a chemical compound called a carbohydrate, the simplest form being glucose, as chemical energy producing oxygen molecules as a byproduct. If you look at the equation below, you can see carbon dioxide and water are inputted in the presence of chlorophyll and sunlight, and you get C6H12O6, which is a glucose molecule, which is the simplest sugar from which other carbohydrates or sugars are produced, and of course oxygen is produced as a byproduct. So where does photosynthesis occur in plants? The primary organ of photosynthesis in plants is the leaf. Some plants might modify the leaf stalk or petiole into a leaf-like photosynthetic organ called a phyllodes, or a stem might take the function of a photosynthetic organ by flattening to form a cladode or chlorophyll, for example, the stems of prickly pears. The leaf as the primary organ of photosynthesis is orientated to present its largest surface area to the sun. Most leaves are also designed to have a large surface area relative to the volume of the leaf to maximize the surface area for the exchange of gases, water vapor, and light energy. Leaves are supported by the stems of the plants and are orientated for maximum sun exposure, except when the plant is trying to reduce water loss through evapotranspiration. Leaves, in their simplest representation, are comprised of a leaf stalk that connects the leaf to the main stem or to a branch, a midrib or main vein, lateral veins, a leaf blade or lamina, and a leaf apex, as well as a leaf margin. There are a wide variety of leaf forms, and you can refer to my video on leaf morphology for a breakdown of the various leaf types. Leaf anatomy. While leaf morphology refers to the external structure of a leaf, anatomy refers to the detailed internal structure of a leaf. All leaves have an upper and lower surface of their lamina or leaf blade, although some highly modified leaves, such as those of succulents, might be quite rounded or bulbous in shape to reduce water loss. The surface of a leaf is covered by a waxy cuticle to prevent water loss, and below this is the epidermis, which is the plant equivalent of the human skin, which protects the leaf from the outside environment. Some leaves may have trichomes or hairs that protect the leaf in various ways by creating a microclimate to prevent water loss, by making the leaf rough and less palatable, or by having hooks or stinging hairs, or even chemicals that discourage herbivores. There's an upper and lower epidermis. Usually on the base of the leaf there are numerous pores called stomata, singular stoma, which allow for the exchange of water vapor and gases such as carbon dioxide and oxygen. You can see the stoma or pores at the base of this diagram. Within the leaf, near the upper surface and just beneath the upper epidermis, is a layer of regularly arranged cells called the palisade mesophyll, and beneath that layer is a layer of irregularly distributed cells of similar composition but more irregular in shape called spongy mesophyll. Both layers of cells contain pigmented organelles called chloroplasts, which regulate or are the site of the process of photosynthesis. The palisade cells are the primary layer in which photosynthesis production occurs. Within the leaf are the leaf main vein and lateral veins, which provide the network of cells, xylem and phloem that transport water and minerals from the roots to the leaves, and carbohydrates from the leaf once they have been synthesized. You can see the xylem and phloem surrounded by the bundle sheath in the center. The stomata. The leaf stomata or pores are surrounded by two guard cells each. These guard cells expand or contract by becoming turgid or flaccid to regulate the opening and closing of each stoma or pore, thereby regulating the loss of water vapor through evapotranspiration and the exchange of oxygen and carbon dioxide into and out of the plant. Chloroplasts are cell organelles found in plant cells that function as the factories for the process of and production of glucose and other carbohydrates during the process of photosynthesis. Each cell can have dozens of chloroplast organelles which appear as oval green balls within the plant cell. Chloroplasts are filled with pigments such as chlorophyll A, chlorophyll B, carotenoids and xanthophylls, which play a role in the photosynthetic process. The color of each of these pigments represents the reflected light wavelengths that these pigments do not use. All other wavelengths that are absorbed are used to provide energy for the process of photosynthesis. The chloroplast consists of a double membrane separating its contents from the cytoplasm of the cell that it occurs in. The chloroplast is filled with a fluid that contains various structures that form an essential part of the chloroplast organelle and which help it to carry out its function. This fluid is called the stroma and is the site of the second part of the photosynthetic cycle, which is called the Calvin cycle or dark reaction. This refers to the fact that the reaction is not dependent on light to take place, but the term dark reaction can be misleading as the Calvin cycle can still occur in the presence or absence of light. Within the chloroplast stroma is a system of double membranes that form disc-shaped structures called thylakoids, which are piled one on top of the other. Such an arrangement of thylakoids is called a granum. The plural would be grana for those. These thylakoids are linked to other thylakoids by double membrane extensions called lamellae, singular lamella. The thylakoids are the sites of the first part of the photosynthetic reaction, namely the light reaction, since light is required for this reaction to occur. The double membranes of the thylakoids separate the contents of the thylakoid, called the lumen, from the stroma. Also contained in the stroma are starch granules, which act as storage units for starch, and lipid globules, which store lipids used to make fats. In addition, there is a loop of DNA and many ribosomes which regulate the production of proteins and which are remnants of the primitive organism that the chloroplast is thought to have been derived from, and which plants came to incorporate. So the thylakoids once again are stacked into a pile known as a granum, and each granum is connected to other grana by means of the double membrane extensions called the lamellae as previously mentioned. Within the double membrane walls of the thylakoids are special protein and pigment complexes called photosystems that direct the light reaction of photosynthesis, which will be discussed later. Associated with these photosystems are other proteins which are part of an electron transport chain and which help with the movement of hydrogen into and out of the thylakoid lumen. Within the fluid-filled stroma, as mentioned before, are the coils of DNA, RNA, and ribosomes, lipid globules, and other contents. There are two main processes involved in photosynthesis. These have already been mentioned, namely the light reaction and the Calvin cycle or dark reaction. So what does the light reaction do? The light reaction, as the name suggests, is the part of the photosynthetic process whereby light energy is used to split water into its component molecules of hydrogen and oxygen in an oxidation process, which then releases two electrons from the water molecule that will form part of an electron transport chain that culminates in the production of NADPH and ATP, two energy storage molecules that will drive the next part of the photosynthetic process, namely the Calvin cycle or dark reaction. So if you look at this, it splits water into hydrogen ions or proteins and oxygen atoms. It releases electrons to drive the transfer of energy in the electron transport chain to enable the production of NADPH. The protons or hydrogen ions are used to drive the production of ATP. About 10% of the oxygen is used for respiration in the plants, and the remainder is released via the stomata. NADPH and ATP are energy storage molecules which drive the Calvin cycle or dark reaction of photosynthesis, and the process occurs in the lumen of each thylakoid. What does the Calvin cycle or dark reaction do? Basically, the Calvin cycle uses the ATP and NADPH produced by the light reaction to provide energy for the production of glucose molecules. This process occurs in the stroma of the chloroplast exclusively, and the process is driven by enzymes. So basically, ATP and NADPH provide the energy for driving the Calvin cycle or dark reaction. It is called the dark reaction because it does not require light to take place, but that does not mean that it cannot occur in the presence of light. The Calvin cycle is part of the photosynthetic process that chemically stores the energy derived from the sun's light into molecules called carbohydrates. Glucose is the primary molecule produced during the Calvin cycle, and it forms the building blocks for the construction of other more complex carbohydrates, which may function in the storage of energy or may play a role as a structural molecule in the case of cellulose in cell walls, or a communication function in cell membranes. Photosystems. The thylakoid membrane consists of two photosystems, which are protein and pigment complexes, and they are also called light harvesting complexes. These are arranged within the double membrane of the thylakoid and are called photosystem 1 and photosystem 2. The associated numbers reflect the order in which they were discovered. However, photosystem 2 is the first protein pigment complex or light harvesting complex involved in the sequential series that forms part of the light reaction. The photosystems are designed to make use of light at two optimal wavelengths, 680 nanometers for the photosystem 2 and 700 nanometers for photosystem 1. The pigments in the chloroplasts that absorb light at these wavelengths are referred to as P680 and P700 respectively therefore. Light photons at wavelengths of 680 nanometers and 700 nanometers strike the numerous pigments such as chlorophyll A and other pigments such as carotenoids and xanthophylls located in these photosystem complexes. This is the impetus that begins the light reaction of photosynthesis within each photosystem complex. Within each photosystem complex are a pair of chlorophyll pigments called a special pair, located in what is known as the reaction core of the complex, which are involved in receiving electrons from the hydrolysis or splitting of water molecules, and which transfer them to a protein called the primary electron acceptor. Complexes and proteins associated with the thylakoid membrane. Apart from the two photosystems, there are other proteins and complexes associated with the light reaction and the thylakoid membrane. These include plastoquinones, cytochrome C, plastocyanin, ferredoxin, NADP reductase, and ATP synthase. They all form part of the electron transport system with the exception of the ATP synthase, which is involved in the movement of hydrogen ions or protons out of the thylakoid lumen and into the stroma. The light reaction, the Z scheme. So what is the Z scheme? The Z scheme is essentially the electron transport chain that shows the path of an electron through the various protein pigment complexes that eventually result in the production of ATP and NADPH. It is called the Z scheme because the electron transport system shows a zigzagging scheme that takes the electrons through the chain as it vacillates between an excited and high energy state to a low energy state as it passes through the various protein pigment complexes. Photosystem 2 has a protein pigment complex known as the light harvesting complex, as mentioned previously. Within this complex are between 300 and 400 chlorophylls and other pigments, as well as protein complexes. These pigments become excited when exposed to photons of light from the sun. The photons, or wave packets, that activate the process in photosystem 2 are in the frequency range of 680 nanometers. The excited pigments have electrons that rise to a higher energy level when exposed to light of this frequency. These chlorophyll pigments then transfer their energy to neighboring pigments through electromagnetic interactions called resonance energy transfer. ATP synthase. The hydrogen ions or protons accumulate in the lumen of the thylakoid, where they will later be used to drive a hydrogen pump called ATP synthase that will use the energy from the process of pumping hydrogen ions to synthesize ATP molecules in a process known as phosphorylation of ADP, which is adenosine diphosphate. The primary electron acceptor, or phaophyton as it is also known in photosystem 2, having accepted an excited electron from the special pair, will now transfer the electron to an organic molecule known as plastoquinone. From thence, the electron will pass through another molecule called cytochrome complex before moving on to plastocyanin, a protein that contains copper. As the electron moves along this chain, it loses energy at each step. Since this energy is used to pump hydrogen ions or protons from the stroma into the thylakoid lumen as the electron passes through the plastoquinone and cytochrome complex molecules. So essentially, the energy that's the electron is carrying is being used to pump hydrogen ions from the stroma into the thylakoid lumen. The plastocyanin protein molecule is the final electron acceptor before the electron passes to photosystem 2, which is next in the sequence of electron acceptors. Whereas photosystem 2 receives its electrons from the hydrolysis or splitting of water molecules, photosystem 1 receives its electrons from the plastocyanin molecule. The electron has lost energy at each step in the electron transport chain and is in a relatively low energy state when it is passed on to photosystem 1. The special pair P700 located in the reaction center or core of photosystem 1 recharges the electron with energy as it accepts the electron from the plastocyanin molecule. The energy used to do this is of course derived from the pigment molecules in the photosystem 1 complex that have been excited by light photons striking it at a wavelength of 700 nanometers. The transfer of energy is once again through resonance energy transfer. The excited electron then passes to a primary electron acceptor known as chlorophyll AO. The chlorophyll AO primary electron acceptor of photosystem 1 then passes the excited electron to a molecule called ferredoxin, and from thence to NADP reductase. The energy of the electron is used by the NADP reductase to synthesize NADPH, binding hydrogen ions to molecules of NADP, moving freely in the fluid of the stroma. In the process of doing this, energy is stored in the bond made between NADP and the hydrogen ion. The NADPH is produced in the stroma and is now made available for the second part of the photosynthesis process, namely the Calvin cycle. After photolysis of water molecules by photosystem 2 in the manganese center, the hydrogen ions or protons produced begin to accumulate in the thylakoid lumen. In addition, hydrogen ions or protons are actively pumped into the lumen as the molecules of plastoquinone and cytochrome complex receive energy as they transfer the electron in the electron transport chain. This causes a buildup of hydrogen ions, creating a concentration gradient across the thylakoid membrane. These hydrogen ions cannot pass on their own through the thylakoid membrane in spite of the concentration gradient. However, a specialized protein in the membrane called ATP synthase acts like a pump to pass hydrogen ions out of the lumen and into the stroma. As the ATP synthase pumps the hydrogen ions out of the lumen of the thylakoid, energy is produced, which is used to create ATP molecules from ADP in a process called phosphorylation. The process is called chemiosmosis since it relies on a gradient of hydrogen ions across the thylakoid membrane. The ATP produced will collect in the stroma to be utilized in the Calvin cycle as a source of energy to drive the cycle of carbohydrate production from carbon dioxide. Linear phosphorylation, photophosphorylation. The production of NADPH through the transfer of an electron of NADP reductase is the culmination of a process called linear photophosphorylation, since the electrons move in a linear fashion from water through photosystem 2 to plastocyanin to photosystem 1 and finally to NADP reductase, the molecule that produces the NADPH. The sequence of linear photophosphorylation is indicated in red in this diagram. Cyclic photophosphorylation. In some instances, the linear photophosphorylation process may be interrupted so that the electron does not get to the NADP reductase, but instead it is diverted by ferredoxin back to the electron transport chain at plastoquinone and then moves through the plastoquinone protein and cytochrome complex, where it once again rejoins the electron transport chain, continuing the pumping of hydrogen ions into the lumen of the thylakoid to continue the process of ADP synthesis. This feedback mechanism is thought to serve three main functions. Firstly, it adjusts the system to compensate for a shortage of NADP in the stroma, which is required for the production of NADPH. Secondly, in plant photosynthetic cells that have high demand for ATP molecules to drive their reactions, for example in bundle sheath cells associated with C4 plants, the cyclic photophosphorylation process provides more ATP. And thirdly, cyclic electron flow may play a role in protecting photosystem proteins against excessive light damage by allowing photosystems to repair light-induced damage. The Calvin cycle. The Calvin cycle has three main processes: carboxylation, reduction, and regeneration. The carbon fixation or carboxylation process involved in the Calvin cycle takes place in the stroma of the chloroplast and uses the products of the light reaction, namely ATP and NADPH, to drive the production of carbohydrate glucose from carbon dioxide. The carbon from the carbon dioxide is used to construct a 6-carbon molecule in a 3-step process. The 3 steps of the Calvin cycle are carbon fixation, reduction, and regeneration. Carbon fixation is the first step and involves the bonding of carbon dioxide to RuBP or ribulose 1,5 bisphosphate, which is a 5-carbon molecule. This process is catalyzed by ribulose bisphosphate carboxylase, an enzyme, and results in the formation of a 6-carbon molecule which is unstable and which immediately breaks up into 2 3-carbon molecules called 3-phosphoglycerate. The 3-phosphoglycerate then becomes part of the reduction cycle. It is produced by a reaction between carbon dioxide and ribulose 1,5 bisphosphate as mentioned, and is now used to produce molecules of glyceraldehyde 3-phosphate by the addition of electrons from NADPH to reduce the 3-phosphoglycerate molecules into glyceraldehyde 3-phosphate. The process starts when 3-phosphoglycerate gains a phosphate group by the addition of ATP to form ADP, and 1,3-bisphosphoglycerate is produced. 6 ATP molecules are used in this process. This molecule is in turn converted into the glyceraldehyde 3-phosphate molecule by reduction with NADPH to form NADP and the molecule itself. 6 NADPH molecules are used in this process to produce 6 NADP molecules. The NADP and ADP return to the light reaction as substrates for the production of ATP and NADPH. Regeneration. Glyceraldehyde 3-phosphate, having been produced by the reduction process, is then used to regenerate ribulose 1,5 bisphosphate and also to synthesize glucose. In a single Calvin cycle, 2 carbon dioxide molecules must enter the cycle, producing 1 glyceraldehyde 3-phosphate molecule which goes to make glucose, and the remaining glyceraldehyde 3-phosphate molecules go to produce a single ribulose 1,5 bisphosphate 5-carbon molecule required to run the next reaction. 9 ATPs are used during the process, of which 3 ATPs are used during the regeneration step to produce ribulose bisphosphate. The cycles required for the production of a single glucose molecule are actually several in order to produce a single glucose molecule. 2 glyceraldehyde 3-phosphate molecules are required to make a single 6-carbon glucose molecule. So it requires 6 turns of the cycle to produce a glucose molecule, using 6 carbon dioxide, 18 ATP, and 12 NADPH for the process to occur. The reason being that 6 ribulose 1,5-bisphosphate molecules are required in order to produce the 2 glyceraldehyde 3-phosphate molecules, which go into the production of a glucose molecule. Here we have a more detailed representation of the Calvin cycle, and you can see the molecular structure of the individual molecules. 6 molecules of ribulose 1,5-bisphosphate start off with the addition of 6 carbon dioxide in the presence of the enzyme ribulose bisphosphate carboxylase to produce an intermediate molecule, carboxylase, of which there are 6 required, which immediately breaks up into 2 3-phosphoglycerate molecules, giving a total of 12 phosphoglycerate molecules, and there are of course 3 carbon molecules. The phosphoglycerate molecule is then converted with the addition of 12 ATP molecules and the enzyme phosphoglycerate kinase to produce 12 1,3-bisphosphoglycerate molecules, and these in turn are converted using the energy from 12 NADPH molecules and the enzyme glyceraldehyde 3-phosphate dehydrogenase to produce glyceraldehyde 3-phosphate, which is the molecule that is used to regenerate ribulose 1,5-bisphosphate, but also to produce glucose molecules. So 2 of those glyceraldehyde 3-phosphate molecules go to the cell cytoplasm, where they are used to make sugars, including glucose, which is the simplest and is a 6-carbon sugar. The other 10 glyceraldehyde 3-phosphate molecules are used to resynthesize ribulose 1,5-bisphosphate, called regeneration, and that requires 12 ATP molecules. Photorespiration. In principle, photosynthesis should be fairly straightforward. If enough light, carbon dioxide and water in the presence of sufficient chlorophyll pigments is available, food in the form of glucose and other carbohydrates can be synthesized by autotrophic organisms to be used for the plant's metabolic needs. A convenient production of oxygen accompanies the process, of which 10% is utilized by the plant and the remainder is released through the stomata of the leaf to be made available to other living organisms, which would use the oxygen in respiration to release metabolic energy stored in carbohydrates in organic bonds. In reality, of course, the resources needed for photosynthesis are not always freely available, primarily because plants may in some cases have to compromise a little to achieve their metabolic goals. For instance, where plants are deprived of water through drought, extremely high winds and temperatures, and through sandy soils which drain too freely, leaving the soil depleted of its precious resource, plants must make an adjustment to compensate for this lack of available water. One such strategy is to reduce the rate of evapotranspiration by closing the stomata during the hottest time of the day. Unfortunately, closed stomata, while effective in reducing water loss through evapotranspiration, also leads to a shortage of carbon dioxide, which is absorbed from the air through the stomata. Carbon dioxide is, of course, the source of carbon, the primary building block for organic molecules. So without enough carbon dioxide, the process of photosynthesis can be halted. Added to this, a buildup of oxygen occurs when the stomata are closed. The oxygen excess can have a profound effect on the process of photosynthesis by interfering with the absorption of carbon dioxide, where it is bound to ribulose bisphosphate by ribisco, the carboxylizing enzyme of the Calvin cycle, to produce the unstable precursor ribulose bisphosphate carboxylase, the 6-carbon intermediate of the 2 3-phosphoglycerate molecules that each step produces. Essentially, at higher temperatures and lower carbon dioxide to oxygen ratios, the ribisco molecule has an increasing affinity for oxygen molecules, leading to a process called photorespiration. So, when ribisco reacts with oxygen in an oxygenation reaction, only one 3-carbon 3-phosphoglycerate molecule is produced, and in addition a 2-carbon molecule called phosphoglycolate is also produced. Phosphoglycolate cannot take part in the normal Calvin cycle, and so it is effectively stealing its 2-carbon atoms from the reaction cycle. Although the plant tries to compensate for this by sending the phosphoglycolate through a series of reactions in other organelles, ultimately it can only retrieve 3/4 of the carbon atoms lost during the period of photorespiration. Ultimately, during normal photosynthesis, 6 carbons are gained in the production of a 6-carbon glucose molecule, while photorespiration, under the same conditions, where 6 oxygen atoms rather than 6 carbon atoms are absorbed, will result in a net loss of 3 carbons using the equivalent amount of carbon resources. So clearly photorespiration uses energy and resources. There are, however, some advantages to this reaction. Firstly, photorespiration appears to provide some protection against light-induced damage to molecules involved in photosynthesis. That is, it has a photoprotective effect. Secondly, it helps to retain the redox balance in cells. And finally, it supports the plant immune defenses. All plants that utilize the normal Calvin cycle reactions are called C3 plants because they produce a 3-carbon phosphoglyceraldehyde atom during the carboxylase phase of the Calvin cycle. These plants are at a disadvantage in high temperature environments, and the resultant process is photorespiration. Some plants, mostly members of the grass family Poaceae, have to maintain production under high temperatures and low carbon dioxide to oxygen ratios. These are called C4 plants because they can produce a 4-carbon molecule during the process of photosynthesis. Once photorespiration has been initiated by high temperatures and low carbon dioxide to oxygen ratios, the plant has to try to retrieve the 2 carbon atoms contained in phosphoglycerate that are lost during its production through oxygenation of ribulose bisphosphate by ribulose bisphosphate carboxylase. This is a relatively involved process and takes place in several plant cell organelles, namely peroxisomes, mitochondria, and chloroplasts. The following steps occur during the process of photorespiration, which produces phosphoglycolate, leading to eventual phosphoglycolate transformation and carbon retrieval. First, there is oxygenation of ribulose bisphosphate to produce 2-phosphoglycolate and then the 3-phosphoglycerate. That would be the normal photorespiration reaction. The 3-phosphoglycerate can then be used in a normal way for the Calvin cycle to continue, and the 2-carbon phosphoglycolate cannot take part in the Calvin cycle and removes 2 carbon atoms from the cycle. However, it is now directed through a series of reactions that occur in a series of plant organelles to eventually be returned to the Calvin cycle as 3-phosphoglycerate. The carbon retrieval process begins with the conversion of the phosphoglycolate to glycolate by the addition of water and the removal of a phosphate group. The glycolate is then transported to the peroxisome, where oxygen is added to convert the glycolate into glyoxylate with the release of peroxide. The peroxisome is a cell organelle that functions in the metabolism of lipids and other hydrophobic molecules, but it also helps process hydrogen peroxide, a highly reactive and potentially harmful molecule produced during the carbon retrieval process following photorespiration. The glyoxylate is then converted into glycine before being transported to the mitochondria, where it is converted to serine. The serine then enters the peroxisome and is converted to a pyruvate, and then finally a glycerate before being transported to the chloroplast, where it is converted into the usable 3-phosphoglycerate by the addition of ATP. The 3-phosphoglycerate can now be used in the Calvin cycle to continue the process of production of glyceraldehyde 3-phosphate for glucose production and regeneration of the ribulose 1,5-bisphosphate, which continues the Calvin cycle. The process is energy intensive and is costly to the plant as it requires additional ATP and NADH to reconstitute the 3-phosphoglycerate. Some plants have adapted to avoid this pitfall of photorespiration. C3 plants are all plants that rely primarily on the 3-carbon phosphoglycerate molecule as part of the Calvin cycle to generate glyceraldehyde 3-phosphate, which is used to manufacture glucose and other carbohydrates. As mentioned already, the danger of high temperatures and low carbon dioxide to oxygen levels is photorespiration, which can rob the Calvin cycle of carbon atoms and cost the plant in energy terms to recycle the lost carbon. Some plants have therefore adapted to cope with high temperatures induced by the local climatic conditions and low carbon dioxide to oxygen ratios induced by the response of the plant to loss of water through evapotranspiration and subsequent loss of the stomata by utilizing a 4-carbon molecule, malate, to store carbon dioxide at night, so that the stomata can remain closed during the hottest times of the day while still allowing the plant to have sufficient carbon dioxide for the Calvin cycle. Plants that have adapted in this way include members of the grass family, Poaceae, and various other fast-growing, sunlight-loving plant species such as primary colonizers. These plants fix carbon dioxide to phosphoenolpyruvate molecules, facilitated by the enzyme phosphoenolpyruvate carboxylase, and these make oxaloacetate, which is in turn converted to malate. The phosphoenolpyruvate carboxylase does not have photorespiratory capabilities, and it has a high affinity for carbon dioxide, making it advantageous over the C3 enzyme Rubisco, which has a lower affinity for carbon dioxide, particularly when oxygen levels are high, and which can facilitate photorespiration. In addition, the oxaloacetate, once converted to malate, is decarboxylated, that is, carbon dioxide is removed or yielded, which is then fed to Rubisco and the Calvin cycle. The process of the 4-carbon pathway is a 2-step process that takes place in 2 separate cell types, the outer mesophyll cell layers and the inner bundle sheath cell layer. This pathway is also referred to as the Hatch-Slack pathway. The mesophyll cells absorb carbon dioxide from air available in the interstitial spaces surrounding the mesophyll cells as absorbed through the leaf stomata. The carbon dioxide is fixed into phosphoenolpyruvate molecules within the mesophyll cells and produces oxaloacetate and then malate. The malate then passes via plasmodesmata, which are connections between the cell walls of the mesophyll and bundle sheath cells, into the bundle sheath cells. Here the malate is decarboxylated so that carbon dioxide yielded can be used for Rubisco and the Calvin cycle. The process does cost more energy than carbon fixation in a normal C3 plant Calvin cycle pathway. However, when temperatures become excessive and the risk of photorespiration subsequently increases, the process becomes much more efficient in C4 plants in comparison to C3 plants. In the process of the malate being decarboxylated to release its carbon dioxide molecules for Rubisco and the Calvin cycle, it converts to pyruvate, which is then passed back through the plasmodesmata from the bundle sheath cell to the mesophyll cell. Here it is phosphorylated by the enzyme pyruvate phosphate dikinase to generate more phosphoenolpyruvate to be fed back into the C4 cycle or Hatch-Slack pathway. It is important to note that the Calvin cycle in C4 plants occurs in the bundle sheath cells of the leaf that surround the transport tissue of the leaf veins, while the light reaction occurs in the mesophyll cells. So the processes of the light and dark reaction are separated in space in C4 plants, whereas CAM plants, which we'll discuss later, are separated in time. All reactions occur in the mesophyll cells and CAM plants, but they are separated in time by storage of malate in the plant vacuoles. Crassulacean acid metabolism, or CAM, is a carbon fixation pathway that was first discovered in plants of the family Crassulaceae, hence the name, which are commonly known as stone crops. The carbon fixation pathway has since then been discovered in the pineapples of the family Bromeliaceae and in various species within the cactus family Cactaceae. The CAM pathway works at night to capture carbon dioxide in molecules of malate, which are immediately stored in a vacuole within the cytoplasm of a photosynthetic plant cell. This allows the plant to keep its stomata closed for most of the day when conditions of high temperature might threaten the plant with water stress or excessive water loss through evapotranspiration, drought, and or low rainfall, or simply sandy soils that drain easily and do not hold usable water. During the day, when the stomata are closed, the plant can still get the required carbon dioxide needed for the metabolic process of photosynthesis in the Calvin cycle by releasing malate from the vacuoles. The malate is decarboxylated, in other words, carbon dioxide is removed to yield the carbon dioxide to provide a steady supply of carbon dioxide for the ribulose bisphosphate carboxylase enzyme in order to produce 3 phosphoglycerate molecules from ribulose bisphosphate. The malate is broken down during this process into pyruvate, which can be used to resynthesize glucose, which can in turn be converted into starch for storage. This is important because at night, during the process of fixing carbon dioxide into malate, there's a high energy demand which requires starch to be broken down into glucose, which provides the energy for the glycolytic generation of phosphoenol pyruvate. Phosphoenol pyruvate is generated so that it can be carboxylated. Carbon dioxide is added to the molecule to form malate, which stores the carbon dioxide in the plant vacuole until it is required during the daytime. Photosynthetic reaction processes, as the light reaction provides ATP and NADPH sufficient enough to run the Calvin cycle, where the carbon dioxide is used. Carbon dioxide is removed from the malate called decarboxylation by a malic acid enzyme, which may be dependent on NADP or NAD depending on the species of plant concerned. The CAM process separates carbon fixation and the Calvin cycle in time within the mesophyll cell. So to compare C3, C4 and CAM plants, C3 plants, which is most plants that are known, have no separation of carbon dioxide fixation and the Calvin cycle. Their stomata remain open during the day and they are adapted for cool, wet to moderately warm environments. C4 plants, which include most grasses, or actually all grasses, which are including the grain crops and many pioneering species, separate in space their carbon dioxide fixation and Calvin cycle processes between the mesophyll cells and the bundle sheath cells. The stomata are open during the day and they tend to be adapted for hot, sunny and wet or dry environments. CAM plants are plants of the family Crassulaceae, some Cactaceae and the bromeliads as well. These are separated in time between day and night within the mesophyll cell. The stomata open only at night and they are adapted for very hot, dry environments. So that concludes the video on photosynthesis. It's a very complex process and I would recommend going over each of the steps in more detail by looking at the video again and skipping ahead to various steps. Thanks for watching. I hope you enjoyed this and you got something out of it. Please like and subscribe. I'd appreciate your support. Thank you.